मॉर्निंग टू यू ऑल। तो अभी हम स्टार्ट करेंगे हमारा फर्स्ट चैप्टर दैट इज़ रिकॉम्बिनेंट डीएनए टेक्नोलॉजी। व्हाट इज़ एक्चुअली रिकॉम्बिनेंट डीएनए टेक्नोलॉजी इज़ वी आर इंजीनियरिंग ए जीन। सो आप पूछेंगे कि इंजीनियरिंग का मतलब क्या है। इंजीनियरिंग इन द सेंस ये है एक डीएनए। आई एम गोइंग टू use this part, take out this part and transfer this to another region, और इस डीएनए के AGTC जो न्यूक्लियोटाइड्स है आई एम गोइंग टू alter it और आई एम गोइंग टू मेक some changes in this DNA. All these comes under recombinant DNA technology. So the present biotechnologies completely depend upon molecular techniques. One of the very important, very promising technique of biotechnology is recombinant DNA technology. In short, we can say that genetic engineering is altering the DNA or making changes on the DNA so as to get some beneficial uses. Always keep in mind biotechnology is related to beneficial use. Let us take an example. Nowadays, insulin is a very common medicine given for diabetic patients. So how do this insulin produce? Earlier it was isolated from pigs, horses, cows. That means we used to slaughter them and take out their pancreas. From the pancreas they used to isolate insulin. It's a hormone, you all know. But to make alive a patient in a diabetic patient for a whole year, we'll have to kill at least 50, 300, 200 pig, horses, cows to maintain the life of a diabetic patient. But this scenario has been changed after the entry of genetic engineering. अभी हम क्या कर रहे हैं, इंसुलिन का जो जीन है, that gene, not the protein, the gene representing insulin, this is there in every human being's cell. This gene is taken out from the individual and transferred to an easy growing organism, that is bacteria, and then this bacteria is allowed to multiply, and this bacteria also start expressing, producing that gene or producing insulin. So in order to isolate insulin from the bacteria, it takes only certain weeks, because bacteria divides in every 20 minutes, let us say 20 minutes. So what happened, for the sake of all these killing these animals, we have transferred the gene from the human to bacteria, and then that insulin is produced in bacteria. This is achieved only because of biotechnology, or only because of recombinant DNA technology. Now let us see what are the objectives of recombinant DNA technology. What are the applications or uses of our recombinant DNA technology? We do recombinant DNA technology for two purposes. Objectives. Either this recombinant DNA technology is to make clone of a gene. Clone means copies, multiple copies. Or it may be used for expressing a protein or a gene. Let us write as gene. Expressing a gene or cloning a gene. There are two objectives. अभी हमने जो इंसुलिन की बात किया है वो क्या है? Expression of a gene. What we have done? We have taken the gene from human, transferred to E. coli, bacteria, and allowed to produce inside E. coli. So made to express a gene in a foreign organism. Insulin belongs to human being, but now it has been transferred to bacteria. So another major objective is cloning of genes. For example, I have a gene A. This gene is being transferred from human to bacteria. And what I am doing, I am making multiple copies, billions of copies of this gene. Just I need multiple copies of gene. I don't want the protein or the expressed product of this gene. I am just making copies of these genes. This is also one of the objectives of recombinant DNA technology. So let us see what are the major steps involved in this. अब हम इसको कैसे करेंगे? Actually, what are the major steps associated with making or making a copy of gene or expressing a gene, whatever it may so. Let us take an example, gene cloning and its basic steps. So first of all, we need a gene, a desired gene, and that gene means in an organism, in its cell, there may be one gene which is having some quality that we need to clone. So first and foremost thing is we have to have a gene of interest. Our gene of interest is also known as insert. Okay, this insert which is containing gene of interest, first step in the recombinant DNA technology is isolation of insert, isolation of the insert or isolation of this gene of interest. Then at the same time we need to have a vector. Vector means a vehicle that can transfer our gene of interest from one organism to another or one cell to another cell. So we call it as vector, cloning vector, and these two need to be jointed. For that we need some molecular tools. आपने देखा होगा कारपेंटर एक लकड़ी के पीस को काट के दूसरे लकड़ी के पीस के साथ जॉइन करते हैं. Same way, we need to precisely cut this gene of interest and we have to fix that gene of interest inside the vehicle so that this vehicle can be delivered to host. So second step is making recombinant plasmid or vector. Recombinant इसलिए हो गया ये, earlier इसका combination अलग था, अब हमने इसमें insert डाला तो ये recombined हो गया. And the third step is transferring this gene of interest to the host. Okay, so हमने recombinant plasmid बनाया और इस recombinant plasmid को अभी हमें उसके नए सेल के अंदर ले जाना है. So next step is transfer of these recombinant plasmid to a host cell. Now from the host cell, or this recombinant plasmid will multiply inside the cell, as well as when the cell multiply, their number also get multiplied. So the last step is selection of the host which is containing our recombinant plasmid and making copies of it. So altogether the recombinant DNA technology or gene cloning is having 4 steps. Isolation of DNA, that is gene of interest, Generation of recombinant DNA, that is by insertion of DNA to the carrier molecule. Third, transfer of the DNA to the E. coli or host cell. Last one is selection and multiplication. Okay, the first recombinant molecule was done by Paul Berg, Herbert Boyer, Annie Chang, and Stanley Cohen in the year 1973. Okay, so this is in short the basic steps of recombinant DNA technology.